So a little bit about Wei. He was actually my graduate student when I was at Caltech. He joined as an assistant professor at ISB in 2018, and he was promoted to associate professor just this past year. And prior to joining ISB, Wei was an assistant professor at UCLA. Notably, when he obtained his Caltech PhD in 2014, he received I can't remember the name of the award, but it was the single award for the outstanding PhD thesis that year at Caltech, a pretty non-trivial award. He's been recognized as an Andy Hill Cancer Research Endowment Distinguished Researchers Award. He received an award from the Chinese government for outstanding science as a student abroad in 2013. We've collaborated for a long time, and he also has his own programs. Much of what he's doing is really at the groundbreaking in terms of trying to think about how not only how cancers and patients respond to various treatments, but trying to figure out, and this is not done very often or very well, trying to figure out what treatments are most appropriate for which cancers. So today he's going to talk to you about a new technology he's developed that provides oncologists with a faster and more accurate way to assign a therapy to a patient. So Wei.
Thank you, Jim, for the introduction. And good afternoon, everyone. And thank you for joining us today. And I'm Wei from SB. It's a great pleasure to share with you some of our exciting research progress in the field of precision oncology. So today, I'm going to talk about a companion diagnostic tool we have developed termed MetaboCore and how it can help physicians to select the most effective systemic therapy for each individual cancer patient. So cancer is a word that strikes fears into all of us. Many people may wonder why cancer is so difficult to treat. One key reason is that cancer isn't just one disease. Every cancer is unique. And even if two people have the same type of cancer in the same organ, their tumors are different. So imagine like two gumball machines like here, they look the same on the outside, but have different gumballs inside. If each gumball represents a cell and different colors represent different types of the cell within a tumor, you can start to see the complexity. This is highly similar to what we see in actual tumors. Tumors are not just made up of cancer cells. They also contain a mixture of other cell types that support tumor growth and survival. Together, these cells create a unique tumor microenvironment for each patient. And this unique environment is why patients with the same type of cancer respond differently to the same treatment. So basically, every patient's tumor is like a lock, and every lock has a unique key. Our goal here is to find the right key for each patient. But the story gets even more complicated as we have an overwhelming number of keys recently, and we need to pick up the one that's fit perfectly. So if you look at plot on the left, it shows the number of FDA-approved oncology drugs from 1950 to now. The first oncology drugs was approved at FDA in 1949, and for decades, we have only about less than 100 drugs available. Then the last decade, there has been a drastic surge in drug development. And in the just past 4 years, from 2020 to 2024, over 250 oncology drugs have been approved, bringing the total number to more than 600. In some way, this is the best of times. Doctors now have more options to treat patients. But it is also the worst of times because this overwhelming number of options make treatment decisions incredibly complex. This actually highlights the desperate need for companion diagnostic tools that can match the right drug to the right patient at the right time. So now you might be wondering, okay, how doctors handle all these complexities when they deciding which cancer treatment to prescribe to the patient. So currently, oncologists mainly rely on the guidelines from the National Comprehensive Cancer Network or NCCN guidelines to choose systemic therapies. These guidelines are based on large-scale clinical trials involving many patients with similar diagnosis, and they continuously updated with the latest data. But while this approach is scientifically sound, it does not account for the unique nature of each individual cancer. And as a result, the effectiveness of a chosen therapy varies, and only a fraction of patients respond well. So with the surge of FDA-approved oncology drugs we just talked about, doctors often face situations these days, where multiple treatment options on the table for consideration, but there are no reliable biomarkers to predict which one will work. So they have to choose the therapy empirically, which I like to call it a polished guess. So take metastatic colorectal cancer, for example. There are two first-line chemotherapy options these days, like called FOLFOX and FOLFIRI, or for simplicity, let's just call them like drug A and drug B. Both have about like 50% response rate, meaning like half of the patient won't respond to either drug. Oncologists often like start with drug A or drug B, right, based on their best guess. They're rolling the dice, and if the tumor doesn't respond within like 3 to 6 months, they switch to the other drug. This trial and error approach means that many patients have to endure unnecessary toxicity, extra cost, and delay in seeking an effective treatment. Clearly, we need a better way to help oncologists identify which standard therapies are unlikely to work for each patient at every point where a treatment decision needs to be made. And there are two major ways to predict treatment effectiveness and guide drug selection. The first approach is based on the tumor genomics. So by performing genomic sequencing, we can decode a tumor's genetic blueprint to uncover its hidden vulnerabilities and then target those molecular weak spots with precision therapy. There have been many efforts in this space, and over the past decade. One notable example is the NCI MATCH trial conducted by the National Cancer Institute, which is the largest precision oncology trial to date, involving 40 different treatment arms and over 6,000 patients. But the results from this trial were rather disappointing. The numbers on the table may be too small to see, but the key takeaway is that the overall response rate, the average overall response rate across all the arms was less than 5%. Okay, and several other precision oncology trials conducted outside the United States involving like over 7,000 additional participants reached similar conclusions. So this disappointing result make us question whether solely relying on genomic information has inherent limitations. And it also drives the renaissance of the functional precision oncology approaches. So why not just perturb the system and see what happens, right? For example, if we want to know if a dog is friendly or not, we can just cautiously poke it and see its reaction. This is how we learn things in our childhood. Similarly, we can take the tumor specimen out from the patient, establishing tumor model and directly testing how this tumor respond to different drugs. This approach will give us a real-world glimpse into their sensitivities and help doctors to choose the most effective therapy. So, like obtaining tumor material, right, and establishing a tumor model and testing drug response seems like such a simple concept. People have been establishing like cancer cell lines for over 65 years. So why hasn't this approach been widely adopted in clinic after so many attempts? What is the gap here? A primary obstacle is actually the limited amount of starting material. Here, for instance, is a picture of a diagnostic needle biopsy. This tiny sample actually represents the amount of tissue doctors take from cancer patients for diagnosis. It's as thin as a fishing line and as light as a housefly. With such a small quantity, it's nearly impossible to perform a traditional drug sensitivity assays, especially when multiple drugs need to be tested. To get around this limitation, researchers have tried to augment this teeny tiny amount of study material by establishing like tumor cell lines or growing them as a xenograft in mice or creating like 3D tumor organoid. But all these methods come at a cost. They compromise the original tumor microenvironment, which is vital for the genuine drug response evaluation. So let me show you what I mean here. So on the left, we actually have a picture of a whole human colon cancer. And the green spots here are cancer cells, and they are surrounded by many other cell types, and denoted by other colors, and all working together to create a complex, heterogeneous tumor microenvironment. And on the right, you see a human colon cancer growing in a mouse. Again, the green spots are cancer cells, but they are now distributed in a much more uniform way across the space. Most of the supporting cells within the tumor microenvironment are gone. So it's like this Gumball machine versus this Gumball machine filled with all right Gumballs. So the stark differences between these two images highlight why so many drugs that give us hope in animal studies lead to heartbreak when they fail in human trials. And without the original tumor microenvironment, we lose the context that make the drug response real. So what we need to do here is to develop a method that allow us to test drug response for a sufficient number of drugs directly on patients' original tumor material without expanding them. This is the driving motivation behind our MetaboCore assay. To make best use of these limited tumor samples for multiple drug testing, we use microdissection to cut those needle biopsies into small disc-like pieces we call discoid. And from a diagnostic needle core, we can generate hundreds of these discoid, each about the size of a grain of sugar. And we can thoroughly mix them and evenly distribute this discoid into multiple trans vials on a microplate for ex vivo drug testing. So here you can see a picture of those small discoids sitting on a trans vial, and here's a zoom-in picture of those discoid. So we treat this tumor discoid with the drug of interest, and the key advantage here is that these discoid maintain an intact local tumor microenvironment during drug treatment, allowing us to capture the most authentic drug response profile of each patient tumor. So let me show you some microscopic images comparing the primary tumor to the microtumor discoid generated from them. The different colors here represent different cell types, and you can see that they maintain a consistent architecture and also the consistent complexity of the tumor microenvironment. This consistency is expected because these microtumor discoid are essentially just small pieces of the original tumor. So to analyze a drug response with such a small amount of tumor tissue, we have to rely on single-cell technology. And we developed this single-cell metabolic phenotyping approach. So basically, after treating the discoid, we dissociate them into a single cells and load them onto a microchip device like here. And the cells are then stained with metabolic tracers and antibodies and imaged using a high-content confocal imaging system. This allows us to measure cellular metabolic activities that are crucial for tumor growth, survival, and drug response, including like glucose consumption, mitochondrial respiration, and oxidative stress for every cell type within the tumor microenvironment at a single cell resolution. So the underlying principle here is that the effective drug treatment that disrupt the tumor growth will inevitably impact the cell's bioenergetics and redox balance. So if there's no change in metabolic activity, it's a strong indicator that the drug isn't working. So how to understand this? So imagine the tumor as this evil Death Star that needs electricity to function, right? And now think of the drug as a missile or proton torpedo, if you like, targeting this Death Star. And you can tell if the attack is successful or not by simply monitoring the electricity use And if the star is destroyed, the electricity use will drop dramatically. And they may even switch to other type of fuels, like back to the Stone Age. So similarly, glucose is like the electricity for tumor cells. And if the treatment is effective, it shuts down the glucose consumption, just like how a successful attack shut down the power usage of the Death Star. So the picture below like showing some like positron emission tomography scans, PET scans of glucose consumption of a tumor. The colors here represent the strength of the glucose uptake. And as you can clearly see in the left panel, for effective drug treatment, as long as the drug works, right, it's reduced the glucose consumption of the tumor to a level that is barely detectable. But in the right panel, right, for ineffective drug treatment, the drug basically has no impact on the tumor's glucose consumption. So you can think of our metabolic core assay as a single cell version of PET scan. It quantifies not only glucose consumption, but also the use of other fuels for each individual tumor cells within the tumor microenvironment. And these metabolic signatures can accurately reveal whether tumor cells are responding to the treatment or not. For example, here we have a lung cancer sample known to be sensitive to cisplatin and resistant to paclitaxel. So those two are common chemotherapy drugs used in the clinic. And the metabolic core assay actually showed a sharp reduction in glucose consumption, as you can see here, and a switch to other type of fuels, and also a significantly increased the cellular stress under cisplatin treatment, indicating it was effective. And on the other hand, the ineffective paclitaxel treatment had no effect on glucose consumption or cellular stress. We can simplify this data into a single indicator here, the glucose consumption to mitochondrial respiration ratio. This makes it easier to see the differences between these two chemotherapy drugs, right? And paclitaxel here does not induce significant changes in this ratio. And the lack of metabolic changes we just talked about is a strong indicator of drug non-responsiveness. So now, let me show you some compelling evidence of how our assay prediction aligns closely with real patient outcome in the clinic. For example, in the case one, basically we have a patient bearing a bile duct cancer, and we collect a needle biopsy from the tumor of that patient and analyze it with a metabolic core assay before the onset of therapy. And we tested for the two first-line chemotherapy regimens, FOLFOX and FOLFIRI. And our result actually indicate the tumor would respond to FOLFOX. You can see the drastically drop in the ratio, but show no significant metabolic changes with FOLFIRI, suggesting that the resistance to the FOLFIRI. So unfortunately, the patient was put on FOLFIRI as their first-line therapy. Here you can see the CT scan before and after the treatment. So after 3 months of treatment, the tumor continued to progress, okay, from like 2.6 centimeter to 3.4 centimeter, consistent with our prediction, but it's definitely not good news for the patient. And case 2 and case 3, which involves like different tumor types and drugs, showed similar results where our prediction matched the patient clinical outcome. So collectively, these examples demonstrate how our assay can transform clinical decision-making, providing real-time guidance that could potentially save lives. And importantly, instead of having patients to risk ineffective toxic treatment for 3 to 6 months, we can provide this information to physicians within just 1 week, okay, allowing them to make informed therapy choices for their patients without delay. Okay, and alongside chemotherapies and the targeted therapies that directly destroy tumor cells, and immunotherapy has emerged as a powerful tool in cancer treatment. It works by harnessing the T lymphocyte, the body's natural soldier, to attack cancer cells. Think of this tumor microenvironment like a gumball machine we just talked about, right, filled with the different colors of gumballs. And here we have a different type of cells, right, packed in this tumor microenvironment. And immunotherapy activates the T cells hidden in this mix, empowering them to launch a direct attack on cancer cells. And its effectiveness depends on whether T cells can be successfully activated by the drug within this complex tumor microenvironment. Then how do we gauge T cell activation? So there are several functional hallmarks of T cell activation, including like the secretion of relevant cytokines and chemokines, and notably elevated glucose consumption and tightly regulated like oxidative stress for sustained T cell active function, and also the increased T cell expansion. All these functional hallmarks can be measured using our metabolomic assay. Here is an example involving a melanoma tumor sample known to be resistant to pembrolizumab, but responsive to ipilimumab. So both are like first-line immunotherapy drugs used in the clinic. So for simplicity, we just refer it as a P drug and I drug. So now we shift our focus from cancer cells to T cells within the tumor microenvironment. And we measure the metabolic signature of T cell activation, basically the glucose consumption to oxidative stress ratio, along with the T cell expansion and the cytokine secretion profiles. And you can clearly see that the effective eye drug treatment acts like a power surge, so boosting the glucose consumption and driving the explosive T cell growth and unleashing granzyme B from T cells, an enzyme akin to a molecular grenade that triggers cancer cell death. And in contrast, the ineffective P drug did not show these functional hallmarks of T cell activation. So after seeing all these examples, now you might be wondering how the metabolic core assay can be used in a real clinical setting. So let me walk you through a typical treatment journey for patients with a colorectal cancer liver metastasis to give you some context here. So for those patients, after the initial diagnosis, usually through like CT scan and a needle biopsy, patients will receive their first-line therapy. And some patients respond well, showing a significant tumor shrinkage, and then may undergo a surgical resection followed by the adjuvant chemotherapy to remove any residual disease. However, the majority of patients will experience a tumor recurrence within 1 to 2 years, requiring a second-line or even the third-line systemic therapy to control the disease until they run out of standard of care options. This entire process involving multiple treatment decisions and drug selections, and this is where the metabolic core can be a game changer. By providing the real-time insight into the patient's unique tumor status right before the onset of each therapy, so metabolic core can help identify effective treatments and rule out ineffective options. So even when a patient, at the last stage, exhausts standard treatment options, and metabolic core can still be useful by evaluating the potential efficacy of novel therapies for off-label use. So basically, we see metabolic core as a trusted companion for both doctors and patients, guiding each crucial decision point in the patient treatment journey and offering hope when it's needed the most. Together with leading surgeons and oncologists, we are on the brink of making this a reality, hopefully within the next few years. So with that, I'd like to conclude this presentation by acknowledging the incredible effort of my team members and Yintang, excellent research scientist in my lab who is primarily working on this project and develop this metabolic core platform, and also our extraordinary collaborators, especially Dr. Raymond Young and Dr. Chuck Dresser, who are leading surgical oncologists treating their patients every day. Their dedication to their patients and their belief in the value that our metabolic core assay have driven them to work enthusiastically with us to try to bring this assay into the clinical space. And I also want to acknowledge the funding agency like NIH, like Andy Hill Care Fund, and Washington Research Foundations. Their support have made this work possible. Last but not least, thank you all for your time and attention today.
Hi, Wei. That was really good and inspiring. And as you might imagine, there are a few questions from the audience. I think one of the theme of the questions is, how do you imagine this rolling out from, where it is in your lab to being a tool that people would use in the clinic?
Yeah, this is a great question. So basically, at the current stage, we are trying to work closely with I would say the target customer that would eventually try to use these tools, like those surgeons or oncologists, and we try to work them closely to basically to develop this tool and try to demonstrate a great predictive capacity of metabolic crisis, to the on the patient clinical outcome. And after that, we were trying to make this move this assay into a clear lab space, and that basically allow us to generate results that can be used in the clinic to inform patient care. And this would be involve like a large scale, basically prospective clinical trial that we can demonstrate, like using our tool compared with the standard of care treatment, we can indeed like improve the treatment precision and potentially lead to a better survival time for the patients. So this will basically if we can successfully demonstrate that in a prospective trial, I guess we're gonna try to seek regulatory approvals from FDA and try to move this move it as a companion diagnostic to into the clinical space. Of course, I'm guessing it may involve a centralized lab, maybe operated as a company or even other type of form, maybe in the local medical center as a core service, and to receive needle biopsies from different requesters, like the surgeons or oncologists who want to know the therapy information, basically what therapy works for a particular tumor type for their patients, and we can try to offer this result back in a timely fashion, so to give us informed to allow them to make informed decision on how they want to treat their patients.
So you're working with both UW and Swedish,
Right.
and Ray Young, and I guess Chuck Drescher.
Yeah.
So when you do roll this out, I mean, you talked about you're sort of doing a retrospective study now. That's basically what, in essence, you're doing. And to do a prospective study, do you need a CLIA lab to do the prospective study, or can you do it?
We are actually now doing a prospective study rather than a retrospective. And current stage, we just piggyback our research needle biopsy to a clinical diagnostic needle biopsy. So basically, when the patient is needed to collect the diagnostic needle biopsy, we just get extra research biopsy from them, and then we make our assay and make the predictions before the onset of their therapy. But at current stage, our assay cannot be used to get clinical decision making due to the regulatory reasons. So basically the doctors just make their own decision on how to treat the patient, and we will test the standard of care therapies the doctor is going to select to treat the patient, and then we will compare our prediction with the patient outcome from in the basically real clinical setting. So yeah, it's a prospective prediction, but it's definitely not like we cannot offer interventions. For example, based on our prediction, we think drug A is definitely better than drug B. Then we could potentially advise the doctor to use drug A, but at this stage, doctor is going to make their own decision on how to treat their patients.
But that's where the prospective study would let you see if you basically can help make a better decision.
Exactly. Yeah.
Yeah. So do you see a question that someone asked is about, whether you would roll this out into major medical centers or into rural areas as well.
Yeah.
And you see challenges with rolling it out in places that are not perhaps like UW or Swedish here in Seattle?
Yeah, I guess that's a great question. So we definitely want to democratize these assays and try to make it broadly applicable to benefit as many patients as possible. And so that's why we kind of envision it as like maybe a service like provided by a centralized lab, and we can receive samples from the and across the entire nation, and everyone who at every place who can do a needle biopsy can basically get extra samples and send it to us, and we can run this assay and to basically do this companion diagnostic test and provide results back to the treating physician.
Oh, is that right? So these samples that you're given, they can remain viable for long enough for such a.
For at least for like 24 to 48 hours, it should be okay. We have tried that. We put a sample into like a kind of a medium buffer that maintain the viability of the needle biopsy, and then we ship it like overnight to. I think you can ship it overnight to basically any place within the states, and then the tissue sample is still viable and has a good quality within at least
Great.
within 24 hours. Yeah.
So one question which started off with, this is very exciting work, so congratulations on that. And it asked, how confident are you that it will be adopted widely? And I suppose I'll ask both sides of that question. So what's the competition out there? Is this a novel assay for doing this, or are there other approaches that might do something similar?
Yeah, I guess this is basically there are also, as far as I know, there are also many companies like outside trying to do the same thing, but with a kind of a different approach. Many companies are working on, for example, tumor organoid, and they basically try to grow out the tumor samples and use that as a material to test the drug response. So our central philosophy is that we were thinking the tumor microenvironment is crucial for determining how the tumor going to respond to the drug, particularly in the field of cancer immunotherapies. Basically, you rely on the T cells and other type of immune cell within the tumor microenvironment to attack the cancer cells. And so we were trying to use this what we call the microtumor or microdissected tumor material to do this test directly on the patient original tumor sample rather than expanding it to organoid. I guess this is kind of a unique stuff we were working on, and some other I think some other groups are also like trying to use this microtumor material, or they mince the tumor, or they just cut it to small pieces to try to do the same type of assay. But most of them are used a kind of for example, a viability as an endpoint for the assay, like in a bulk level. And we were thinking the unique thing of our assay is to actually try to use a single cell approach to really get the cellular response at a single cell level. You can not only know how the tumor cell going to respond to the treatment, you can also know how, for example, the immune cell component within the tumor microenvironment, how they're going to respond to the treatment. So this is crucial for like assessing, for example, the immunotherapies regimens, like to see whether the immune cell can be sufficiently activated right within the tumor cell microenvironment after like ex vivo treatment. So this is actually, I think, a kind of a unique feature of Metapore. It can provide a single cell resolution readout of every cell type within the tumor microenvironment that allow us to get like a rich single cell data, and allow us to use, for example, AI or machine learning based approach to really figure out the best predictor, for patient outcome.
Yeah, so I will say that I agree with you that an organoid model is not going to be nearly as good as actually looking at the tumor as it comes out. There's a question about the tumor that comes out that I think is pretty relevant too.
Mm-hmm.
And that's that you've got a pretty small needle biopsy, and oftentimes tumors are really quite big.
Yeah.
And so, how do you think about, and sometimes people will do biopsies, they'll actually take multiple needle biopsies of the same tumor.
Right.
Try to get a better sampling of the whole TME.
Yes.
Is that how you're thinking about it, to get the whole TME out of maybe multiple biopsies if needed, or
Yeah, I think this is a great question. This is also a common challenge in this field, actually. How do you use the small amount of sample to recapitulate, right, the complexity of the original tumor? So basically, because the tumor is highly heterogeneous, and if relying on a single biopsy may get kind of a best result, but it's impractical to get like so many biopsies because it's such an invasive procedure. Our current strategy is we try to collaborate with the radiologists who really help us to get a biopsy from the cancer patients to get needle biopsy from three separate locations of the tumor, and then we just get those three biopsies and make a that macro dissection to form to produce the small discoids, and then we randomly mix them and then evenly distribute them across different test conditions. In that way, we can try to mitigate the influence of the spatial heterogeneity and like
Yeah. That makes sense.
yeah.
So here's a question. And I think this is related to the kind of dense data content that you can collect with single cell imaging
Right.
and the various tests you're doing. Do you use AI or similar computational analysis techniques to sort of mine through the spatial data that you're generating, maybe to draw some broader conclusions?
Yes, this is currently what we are working on. We try to use advanced machine learning models, including the most recent like AI models, to try to use that to harness our rich single cell data set to make a correlation with the patient clinical response. And this requires a kind of statistical sufficient number of patient samples. And we are in the process of generating this type of data. And currently in my presentation, you can actually see some very simple metabolic indicators, like, for example, the glycolysis to mitochondrial respiration ratio can already make a fairly good prediction in terms of patient outcome. But I believe, with the richer data set we are going to get, and by using the machine learning model, that could eventually provide us a much more robust predictors and of patient response. And this is something we are basically working on. Yeah.
Sure. So here's a question about the data that you generate, and is it publicly available, or when would it be? Obviously ISB, we make everything publicly available when it's gone through peer review, and I'm guessing the same is true for you.
Yes, definitely. We are gonna try to publish our findings as soon as possible, and definitely when we publish the result, everything gonna be available. We're gonna deposit the data into the database. Yeah, everyone can use it, even for like secondary analysis.
Yeah, I mean, I think that question and the answer goes to the fact that the independent reviewing process can be kind of useful with an analysis like this, because the reviewers may come up with ideas that you haven't thought about. So when you publish something, you have a little more confidence that it's gone through independent eyes, and then people can begin using it for their own means.
Exactly.
So here's a question. I'm not sure if you're in a position to answer it, but is there a tendency to overprescribe cancer treatments and too quickly?
Yeah, I guess maybe I'm not in a good position to answer this question because I'm not an oncologist who are actually treating those patients. But from what I understand from those my communication with those the practicing oncologists, they really want to know the answer of which drug they should use, because there are so many FDA-approved drugs that are currently available to treat for example, the same diagnosis, and which drug actually give the best efficacy. If they can know it ahead of time, that will definitely save patients time, money, and more importantly, it's gonna provide the most effective treatment for their patients. So that is, I guess, our technology could potentially help to make the treatment decision more precise, and then that actually at the same time try to reduce the over treatment, right? And associated like the unnecessary toxicities the patient have to endure.
Yeah, I think that's, I mean, most tumor indications have got a standard of care type of treatment regimen that physicians will go through. It doesn't really matter where you are, although some physicians may be a little more aggressive with that treatment than others. But the bottom line is oftentimes those treatments don't work. And whether it's standard of care or not, everybody is somewhat unique, something that we really emphasize here at the ISB. And so trying to really explore that uniqueness of individuals to tailor treatments for them in particular is that should be the standard of care. But the technology, just as you have talked about today, is just not getting there.
Yeah. Right.
So I guess the last question that oh, hold it, there's more. They keep coming in. So what led you to dream up this? I know you've worked on tumor metabolism for a long time and spatial imaging and what have you. So what led you to dream up this approach?
Yeah, I guess I think there's a kind of a urgent need in the field about how to prescribe the right drug to the right patient at the right time. This is basically the whole point of the motivation of the field of precision oncology. So we know there's a very exciting thing, many exciting things happen in drug development field, and we submit more and more drugs keep kicking in to the market. But it's really a kind of an unmet need for the physicians to decide which therapy they really need to prescribe, which therapy will again benefit the patient most. So I was thinking I'm kind of very interested in this precision oncology field, and I'm especially believe kind of integration of genomic-based method and functional testing would make a difference in that, because most of the previous effort in this field have been based into the tumor genomics. People were thinking, if we know the genetic blueprint of the tumor, we'll know everything. We'll know how to treat it, the cancer. But it's turned out not to be that easy. And I think because we have been working on those functional assays for a long time, I think integrating our functional assays with tumor genomics would make a big difference.
Yeah. So when there's actually a number of interesting questions here. I'm trying to parse through them so we don't get too repetitive. So maybe, what do you think is the biggest limitation if you were going to talk yourself out of using this technology, which I don't think you want to do, but maybe talk about where you think the limitations are.
So yeah, that's a great question. I think one limitation we have already mentioned is actually the influence of tumor heterogeneity. Although we are trying to get needle biopsies from a different location, but we are limited by the practicality, that we can. For example, in the clinical practice, we can get, for example, at most 3 needle biopsies. Otherwise, it's going to be too invasive procedure for the patient. So that whether we can use those 3 needle biopsy to get the best answer, whether it can be sufficient enough to represent the complexity of the renal tumor, is still a question mark. That is something we are working on. And the second thing is that we try to harness the tumor microenvironment, and we try to retain that tumor microenvironment and as best as we can during the drug treatment. And this is the key advantage of this technology. But still, is the ex vivo treatment and the in vivo treatment are different things, right? In the in vivo system, you have the systemic circulation, and your drug pharmacokinetics and the pharmacodynamics of the drug profiles are very different from the ex vivo treatment. In ex vivo setting, you basically you treat tumor tissue, by putting the drug in the medium is basically a passive diffusion. So whether that could be good enough as a model, right, to really recapitulate what happened in vivo is also something we are trying to figure out here. But the initial result looks pretty promising, and hopefully, there's no perfect model that can 100% recapitulate what happened in the actual patient in vivo. But still, we hope our model would be sufficient to capture what we need to capture here.
Yeah. So here's a simple yes or no question. I'm pretty sure the answer is yes from what I know about what you've been doing. But you're actually looking at, I mean, you are taking patient tissues from the operating room and looking at them, right? This is so this is you're simulating what would happen if this was actually a tool.
Yes.
Yeah. Okay. So here's an interesting question, one that's after kind of my own heart. How soon after starting treatment can changes in glucose consumption be detected? And how
Yeah.
does timing correlate with clinical outcomes?
Yeah, this is a great question. Actually, yeah, the timing frame is I think depends on the context of like what drug you want to use and what tumor types. But normally, if the tumor respond to a drug treatment, it shut down the glucose consumption very quickly. And definitely, Jim, you know that you are working on this on the same stuff before, like and basically sometimes for effective treatment, for example, targeted therapy or chemotherapy, they can shut down the glucose consumption like as soon as within like 12 hours or even
Sometimes within 3 hours.
yes, or even sooner. Yeah, it's a pretty, that is the reason we use metabolic readout rather than the viability as an endpoint for this assay because it's so sensitive and it's very fast. And you don't have to wait for a long time to tumor cell to die in order to tell you whether the therapy will work or not. You can get a pretty fast, readout of whether the therapy is effective or not effective.
Yeah, and then a related question, which I think also is kind of close to my heart, is if you have a metabolic shift, such as a glutamine or lipid metabolism or something like that,
Yeah.
How reliable is glucose consumption as a marker of treatment success? If you, I think it, I'll give a bit of an answer to this, but maybe you can answer it more because you've been studying this.
Sure.
I suspect that when you see glucose consumption drop, like you can see in maybe a matter of hours, suggests that your drug has hit the target. That's pretty clear. It doesn't mean you've had treatment success.
Right. Right. That is absolutely right. Because in the real clinical cases, what we saw is that for some cases, you can eliminate most of the, for example, 90% of the tumor cells, but the 10% of tumor cell, they become dormant. So they indeed, in those tumor cells, you shut down the glucose consumption and they may switch to other type of fuels like glutamine or fatty acid to maintain their survival. But they kind of become a kind of a quiescent cells. They you in the clinic you can what you see is from a CT scan you see a partial response, the tumor shrink, but there's still a minimal residual disease there, and those are kind of a reservoir for the tumor recurrence. So I think this is also one exciting potential of these technologies is that it not only can report whether how the majority of tumor cell respond to the therapy, it can also actually pinpoint those quiescent drug tolerant populations and what the metabolic profile and they have. And I guess that is also related to how you predict the patient outcome, because even though you eliminate 90% of the patient tumor, but if they still have like a small amount of minimal residual disease that cannot be eliminated by the drugs, then eventually it's going to lead to the tumor recurrence.
And there's almost always a minimum residual disease for many of these treatments. Yeah.
Yeah.
So any final comments you want to make, or
we have a high hope on this technology, and we were just trying to try our best and to collaborate with physicians and those practicing oncologists and try to really figure out all these type of the technical hurdles inside this technology, as we just discussed many exciting questions from the audience. Actually, your questions, some of the questions really pointing to the potential limitations or potential technical hurdles we have to get around in order to push this to the clinical space. But our hope is that we can really move it into a clinical space and within like 5 years, and hopefully it can start to create patient benefit, help and like to really deliver a functional based precision oncology assay that help physicians to decide which drug they should choose.